We've talked about zinc here a couple of updates ago, but the story's not done. It's kind of an exciting story that gives me some cautious optimism in terms of COVID-19. But to understand this better, we've got to go back and look at the molecular biology of the cell once again. Here is the nucleus of the cell. In the nucleus of the cell is the DNA. The DNA is a double-stranded string of nucleotides, which are the codes. Those codes are transcribed using RNA polymerase into RNA. That RNA then goes out of the nucleus, gets a 5' cap, and gets a 3' poly A tail, and it's ready for ribosomes to come on to read that code. And that code is then translated into the code of amino acids, which amino acid after amino acid will turn into a polypeptide, which turns into a protein. And proteins are how the cell gets things done. So it moves by proteins, actin and myosin. It can bind oxygen through hemoglobin. It can do cellular metabolism. All of those enzymes in glycolysis, in the citric acid cycle, all those things that you learned in biochemistry, those are all enzymes, those are all proteins. So that's the normal situation. Now enter coronavirus. Coronavirus has its own genome. It is made out of RNA, and that RNA just happens to have a 5' head and a poly A tail. So when it pops into the cytosol, it's going to be read by those same ribosomes that can't tell the difference. Except this time, instead of making a protein that's useful to your cell, this RNA that comes out of the virus is going to make something called a RNA-dependent RNA polymerase. And it's this enzyme right here that is going to read from the 3' M to the 5' end of the viral RNA and replicate it. So this RNA-dependent RNA polymerase makes more viral genomes. It's also known as replicase for that reason. And there's something that has been shown to inhibit this replicase, and that is zinc. Zinc will shut down RNA-dependent RNA polymerase or replicase. And so that is what we learned. The problem is, how do you get zinc inside the cell? The problem with zinc is that it's an ion. It's a 2+ ion. And ions cannot get through the cellular membrane unless there's a transporter that allows it to come in. In fact, the way that they tested this in the paper is with an ionophore, which allows the zinc to come into the cell. So they could see that the activity of this RNA-dependent RNA polymerase was reduced. This is the paper. Zinc inhibits coronavirus RNA polymerase activity in vitro, and zinc ionophores block the replication of these viruses in cell culture. When they looked at the SARS-CoV virus, that was the one that was seen back in 2002, as the zinc concentration inside the cell went up, you can see that the byproduct of the RNA-dependent RNA polymerase went down, 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 clearly demonstrating that zinc intracellularly is going to block this very important enzyme of the virus. Well, that's great. We've got zinc that's going to block it, but how are we going to get zinc inside the cell? It's one thing to say that you're going to take zinc supplements, but how do those zinc supplements, first of all, get absorbed into your body, into the blood, into the extracellular space? And then finally, how are you going to get that zinc from the extracellular space into the intracellular space in the cytosol where it needs to work on these infected cells and these viral proteins? Well, that's another thing altogether. What you need is some sort of ionophore or some sort of gated mechanism to open and to allow that zinc to come into the cell, increasing the concentration of zinc into the cell so it can block RDRP. Getting back to our idea here of what's going on in molecular, we've got zinc here that needs to shut down RNA-dependent RNA polymerase. And what we're saying here is that chloroquine is going to somehow open up this ion gate and allow the zinc to come in to block and shut down viral replication inside of the cells.